Am I audible? Hello. Am I audible?
Yes, ma'am.
Um, so thank you for joining this session. I'm Fenita Hepsiba. I'm a PhD scholar from IIT Madras. So I will be conducting the live sessions for the NPTEL course, which is Plants for Bioprocessing. So as you all know, this is a theoretical course. Um, so as we progress through the following weeks, we will be able to solve some logical reasoning and questioning type of problems. Um, okay. So we'll wait for a few more minutes because there are a few more people joining. We will wait for another 2 to 3 minutes and then we will start with the session. So let's begin with the session. So um, I would like all of you to introduce about yourself. Where are you from? Your name, where are you from, and your area of interest and what you are doing currently. One by one, please introduce yourself. As this is our first class, please introduce yourself.
Yes, ma'am. Hello.
Yeah, hello. You're audible.
Yeah. Yeah. Myself, Fausia from Kerala. I'm doing research now. This is the first stage of my PhD.
Okay.
Just uh, now, I'm going to do in plant biotechnology.
Okay. Good to hear.
Thank you, ma'am.
Jay, Jay Chitole. I don't know how to pronounce your name. Can you introduce yourself? Hello. Okay. Ms. Pallavi, can you introduce yourself?
Myself Pallavi. I'm from Bangalore, studying currently in Christu Jayanti, final year BSc in Biotechnology and Biochemistry.
Okay. Good to have you here, Pallavi.
Thank you, ma'am.
Um, Harshil, I'm not able to view the full name.
Yeah, hello,
Yeah.
ma'am. Am I audible?
Yeah.
Yeah, myself, Harshita. I'm currently pursuing my BTech in NIT AP
Okay.
in biotechnology.
Okay. Sarah.
Ma'am, myself Sara Jiji from Christy Jayanti College, Bangalore, and I'm studying BSc Biotechnology
Okay.
and Genetics.
Okay. Shreya.
Hello, ma'am.
Yeah. Hello.
Ma'am, I'm Shreya from Chandigarh, currently staying at Chennai. I'm pursuing my third year of BTech degree in biotechnology with specialization in genetic engineering
Okay.
at SRMIST.
Oh, SRM. Okay. Shweta.
Good evening, ma'am.
Yeah. Good evening.
I'm myself Shweta Singh. Currently, I'm living in Maharashtra and I'm pursuing my master's in biotechnology from College of Arts, Commerce and Science.
Okay. Somesh.
Ma'am,
Hi,
myself
Somesh.
Somesh from Central and myself Somesh from Central Tribal University of Andhra Pradesh.
Okay.
Currently studying in third year of BSc Botany.
Okay. Tejashree.
I'm Tesh. I'm doing UG BTech Biotechnology at Satyabama University.
Okay.
Second year.
Okay. Ushma Mansoor. Ushma. Okay. Vaibhavi.
Good evening, ma'am.
Good evening.
My name is Vaibhavi Mengre. Currently, I'm studying in last year of Masters in Biotechnology from University of Mumbai.
Okay. Komalavalli.
Hi, ma'am. I'm Komalali. I'm currently pursuing third year of BSc Horticulture in Tamil Nadu Agriculture University, Coimbatore.
Okay. Pradnya. Pradanya.
Yes, ma'am. Ma'am, my name is Pradnya Chandradre. I am doing pursuing MSc Biotechnology. Who's aspiring MSc Biotechnology from Chennai.
Okay. Swapnali.
Myself, Swapnali Shinde. I am from Maharashtra.
Okay.
I am currently pursuing my master degree
Okay.
from Pillai College.
Okay. Then Divya.
Hello, myself Divya Rao. I am currently pursuing Masters in Biotechnology from University of Mumbai.
Okay. So thank you for introducing yourself. If anybody is left out in introducing. Okay, so I will brief out the how this live sessions will be. So basically, for the 8 weeks, you will be handling assignments, right? So I will be solving similar assignment questions, which will be covering the portions which you have come across in the week one content. So I would like this session to be more than as we are dealing with theory here. So I hope you are going through the sessions as soon as possible. It's been released because it's been released on Monday means our session starts at Friday. So I hope that you guys are reading or listening to the lectures and able to solve the problems and also in the sense the questions given in the assignment. So for this theory session, I would like to um, encourage you guys to learn certain topics, and if whenever you get doubt on those topics, you can ask those doubts in this live session itself. If at all not possible in the discussion forum. And also, one more thing, you cannot discuss about the current assignments here in this session. You cannot ask the answers for those questions here in the session. Okay. So I will be covering the first week's content along with some prerequisite questions which I think you might know already. It's okay if you don't know. I think most of you are from BTech Biotechnology or BSc Biotechnology, right? So if you have any doubts regarding um, bioprocessing and stuff, you can always reach out to me. I will at the end of the session I will be dropping out my email ID. Um, so and one more thing I wanted to make sure that before you come to this live session, listen to the lectures and then come so that if any question arises in your mind about the lecture, whatever you heard or when you go through the lectures, when you get any doubts, you can ask me here. Okay. So this will be more of like an interaction session. But this week, I think I'm meeting you guys for the first time. I will be giving you some assignment kind of questions, and I encourage you to answer them. Um, and also, you can, if as you can ask me whether if you want to discuss any kind of topics here from the upcoming sessions. Okay, so let's go into the questions. I think most of it is prerequisite problems. It's not problems, I'm talking about theory, right? So it will be mostly fill in the blanks or match the following kind of questions. And if you have gone through the videos already, you will be able to answer it, I guess. So I want you guys to answer them. These are very uh, basic questions which you all might know, will be knowing, right? So the question one is, which among the following are distinctive features in the answers? The option A is cell wall, B is chloroplast, C is ribosomes, D is nucleus, and E is vacuole. So any one of you, can you answer? You can even put it in the chat box if you feel uh, you're not able to talk actually. So the question is, which among the following are distinctive features in plant cells? So cell wall, A, B, and E. A is cell wall, B is chloroplast, E is vacuole. So I think cell wall won't be coming because cell wall is present in all the cells, right? In prokaryotic cells, in animal cells, etc. And ribosomes is also there, and nucleus is also there in everything. So I think the answer would be, the correct answer would be chloroplast and vacuole, both of them. Okay, the answer would be vacuole and chloroplast, which are specifically meant for plant cells, and these are um, distinguished compared to other animal cells or which prokaryotes which have the cell wall, ribosomes, and nucleus, right? So the next question is, which is the most commonly used plant basal medium for the culture of in vitro plant systems? What do you guys think of this question? Which is the plant basal media which is commonly used for in vitro plant systems, for culturing of the in vitro plant systems? D. Yes, it is molasses and skog medium. Why it is called as molasses and skog medium? Any idea? I think everybody has worked with molasses and skog medium. Why it is called so? It is in their name. There itself the answer is there. It was those two who has what they had made that media. That is why. Yes, Fauzia, you are right. It was named after them who made that media. Basically, what does the molasses and skog medium contains? Can anybody tell? Not molasses and skog media, just tell what a plant basal media will contain. Everybody has gone through the lectures, right? Guys, this is a theory course. So, what and all a plant basal media will contain? Say, for example, animal cell media will contain serum, antibiotics, and those stuff. So, for plant basal medium, what will be the contents which will be necessary? So, you can start like it needs a food source, right? So, a carbon source is necessary, a nitrogen source. What else it needs?
Growth regulators.
Yes, correct. Growth regulators. So, and others like micronutrients, macronutrients, those all will come into picture. Okay. So, molasses and Scoop has formulated a media which contains uh, what you call carbon, nitrogen, micro, and macronutrients, everything. Um, so there are some messages. Yes, auxins and kinetins. These and all will be plant growth. These are plant growth regulators which will be added depending on the species specificity of the plants. Okay, we'll move to the next question. So, in which phase of growth are secondary metabolites generally produced? Lag phase, active log phase, stationary phase, or death phase? B. Is it so? What is lag phase, log phase, and stationary phase? Can anybody explain? Harshita, C. Yes, the answer is C. Stationary phase. Why it is so? Why you guys are getting confused with log phase and stationary phase? So what happens in the lag phase, log phase, stationary phase, and death phase? It is very similar to your, these phases are very similar to your bacterial cells or any microbial production. But what happens is, yes, Komalavalli is typing. So lag phase is preparing for cells to multiply. Correct. Log is active multiplication. And stationary phase is where what happens? Okay. Next week I won't answer for that. And so today I will explain something like how it will be, how it will be like this now. So here the cells will be preparing for growth. Here they will be actively multiplying. This is your stationary phase and this is your death phase. Over time, how the biomass changes. So X here means the growth or biomass. So what happens here when it is actively growing, this will be basically primary metabolites. And when they are in a stationary stage, they will be producing secondary metabolites. So what do you mean by primary and secondary metabolites?
Primary for their own needs.
Yes. Secondary?
Secondary is like protective or protective activities.
Yes. So when they are growing, generally it will be primary metabolite. So the production would be when they are in what you call, when they have reached a maximum growth phase, then they will be compromised, right? So some will be in death phase, some will be in growing phase, somewhat the death and the growth will be balanced out, and there will be more stress. What happens is like these plants will be producing secondary metabolites when they are under stress or for defense purposes. So that is the reason stationary phase only secondary metabolites are generally produced. If your targeted metabolite from your plant, whatever you are working with, is primary metabolite, then you should select the plant when it is in the log phase itself. But if in case you are working with a secondary metabolite, you should be taking it from the stationary phase of growth. Okay. Later on sessions, you will be able to understand this bell-shaped curve pretty well. So this is a basic question where which is a well-known cryoprotectant. Among these, which one is the well-known cryoprotectant? So what is cryoprotectant first? Glycine. What is glycine? First tell me what is cryoprotectant.
Means to protect the plant cells at
Plant
certain
cells?
temperatures.
From? Freezing. Yeah, from low temperature protection, right? So which one is commonly used? It's not glycine.
Is it methanol?
No, methanol is an extractant, which is a solvent. Yeah, it's a glycerol because toluene and methanol is what? They are organic solvents, right? Glycine is it amino acid? Yeah. So what else is left? It is glycerol. So the answer is glycerol. So what else is added while people generally do cryoprotection? Don't talk about plants general purpose you tell. Does anybody know? Liquid nitrogen, okay. Okay. So there are some few other cryoprotectants also that you will be in the further classes you will be able to understand those better. So what mode of reactor operations can overcome product inhibition? Do you guys are familiar with bioreactor and bioprocess? Everybody? No. Okay. So I no. So if you guys where we are talking about bioprocess, um, okay, I will just give a gist of that. But if you are really interested in bioprocess and bioreactors, you can enroll to the next semester course, which is called the bioreactor design and analysis. That course will be able to give you a very brief idea about bioprocess. So here I will explain each of those, and then if whoever knows the answer can answer it. And here in this course also we cover a bit of this plant cell based bioprocessing. So no need not worry. You will be able to learn those also. So guys, who knows about the bioreactors or mode of the reactor operations? Can anybody brief what is batch, fed batch, and continuous? Nobody wants to brief. Okay, so batch culture is when you are dumping all your input inside and let it sit or run for some time in a well mixed condition, and then after a certain period of time you're taking things out. That is batch. Continuous is what you're giving something in at the it is getting mixed in a well mixed reactor. Your input or your cells, your here your biomass will be there, which is plant cells. You're giving media and you are giving agitation for it to circulate the media well, the same way as in microbial bioreactor conditions. But here the shear will be more, and continuously some output or something is being removed from it. That is continuous. So fed batch will be what? It will be BTech biotechnology. I heard somebody was from BTech biotechnology. Can you explain what is fed batch? Added when? So fed batch, then what is the difference between fed batch and continuous? Okay, so what is fed batch is actually it will start as a batch where you will add everything inside and keep it aside, and then you will started adding your nutrients when there is a depletion of nutrients happening. So you will start externally adding it, but don't remove anything from it. That is the simplest form of explaining fed batch. So where do you think this comes? Why, if there is a product inhibition, which among these you will be using? Any idea? C, right, Kavita? So the answer is C, because if in case your product is inhibitory to your cells or inhibitory to it is causing damage to the cells, then it needs to be removed continuously. So that is why continuous is preferred if in case the product you are working with is being is inhibiting your cells growth. So continuous will be used. For the answer of this question is continuous. Okay. So don't worry, people, if you don't know about batch, fed batch, continuous, it's okay. In your further lectures, you will be given a brief lecture on these batch, fed batch, and continuous process. Okay, then you will be understand. This is a prerequisite. If you guys are not able to answer, it's fine. In the following weeks, you will be able to understand it better. So the next question is, Which bacterial species is called as the natural genetic engineer of plants? Answer A is Escherichia coli, which is E. coli, Pseudomonas bacteria, Agrobacterium tumefaciens, and Staphylococcus aureus. Okay, everybody is pouring in answers Agrobacterium tumefaciens. Okay, any one of you explain why it is Agrobacterium tumefaciens? Can anybody explain? Explanation need not be a bigger paragraph or something.
Because it has a region known as DNA, which with that like if we engineer that DNA region, if it becomes a gene of our child, then like pretty much like a molecular plant.
Okay, your answer is correct. Your way of understanding it is correct. The question was natural genetic engineer of plants because they are known in nature, they are known to cause a disease. What was that? Yes. So it has the ability to transfer its whatever Shreya was explaining was absolutely right, where this bacterium can transfer their tDNA into the genome of the plant cells. So they make a what you call a tumor kind of growth on the surface of the trees or something. So that's why it is called natural genetic engineer of plants, from where we have adapted the technology to transfer anything our gene of interest. Okay. So the next question is which impeller is mostly used for mixing due to large shear forces generated in the fermentation broth? So for people who don't know about impellers, impeller is kind of a mixing tool. It is similar to your whisk, which you use to beat cake batter, everything. How we use it in household, but except that it is for a bigger vessel. Say for example, 500 liters, 10 liters, 11 liters, like that, or 2 liter, 1 liter vessel also. Vessel is something like this, and impeller will be like this. Anything which is a moving part which aids in mixing of liquids and solids inside your reactor. Okay, so here the question is which impeller is mostly used for mixing due to large shear forces generated in the fermentation broth? So what would be the answer? So yeah, Russian turbine. Okay, so how it will actually look? It is a bladed impeller. It will kind of disc shape. It will look like that. If you put it like this, it will look like this. You can add 2 or 3 impellers on the shaft. So this is how it will look. The blades will be angled, so it will push the liquid around more easily and will reduce your 1 minute. Sorry. So that will reduce the shear stress on the plant cells. So kind of easy question. Aggregation of plant cells in cell suspension culture can cause mass transfer limitation at high cell density. The statement is true or false? What is aggregation? Aggregation is basically sticking together. So instead of cells, say for example in microbial cells, if you put them in a flask, they will be completely dissolved. like you will not be able to make out, right? But in case of plants, they will form aggregates like this. They will not be, unless you do some human intervention or changing the media composition, the cells will be mostly aggregates. So the question is, if the aggregate is present in the cell suspension culture is liquid culture, then will it cause mass transfer limitation at high cell density? When the cells are higher, when the biomass of cells are higher or larger, the presence of aggregates, will it cause mass transfer limitation? Mass transfer limitation is what? Mass transfer limitations, or to simplify, I can tell the transfer of oxygen from the broth or the liquid to the cells, that we can assume like that. What would be the answer? If the presence of aggregates, will it cause mass transfer limitations? Is it true or false? Okay, the answer is true. Indeed, the presence of aggregates here, say for example, instead of your cells being single like this, if it is aggregate like this, from the liquid which is here, and your oxygen, say for example, is present, and nutrients also, it is difficult for it to cross this and go into this cell. Correct, no? So if there is aggregation happening in the plant cell suspension, then it is indeed will cause problems of mass transfer, especially at high cell densities. So which of the following reactors are known as pneumatically driven reactors? Stir tongue reactor, bubble column reactor, airlift reactor, or all of the above? So for some people, pneumatically driven means which doesn't have any moving parts. For example, the stir tank reactor will contain impeller, like I told before. Bubble column will be looking like this, where the movement or the nutrient flow and the mixing of the salt, mixing of your media, cells, everything happens by bubbles. Here there will be a sparger, there will be constant bubbling happen. Because of that, there will be mixing happening here. And airlift reactor, instead of bubble here, there will be air, continuous supply of air, which will push and cause mixing here. So this pneumatically means no moving parts for mixing. So now you tell, I have almost briefed out everything. So now you can easily make out the answer here. So now you tell which of the following reactors are called pneumatically driven reactor. If it is stir tank, bubble column, or airlift, what will be the answer? C. Okay. Only C. B and C. Correct. Ashita is correct. So both the bubble column reactor and airlift reactor doesn't contain any moving parts inside it. Say it doesn't have any particular part inside it to cause any mixing here. So these two will be the answer. Okay. And in the question itself, it is asked in plural, right? Which of the following reactors are called as pneumatically driven reactors? Okay. So I think you guys would have studied immobilization somewhere in your degree course. Have you guys come across immobilization? Anybody know what is immobilization is? Immobilization. Okay. So immobilization is entrapping something in a gel-like matrix or something. you would have come across agarose beads, kind of, which you use to entrap your cells to increase its. You can tell, you can actually use them for artificial seeds. It's not an artificial seed, it has some other name. Any cell you can entrap it inside a matrix, gel like matrix, that will provide, that will actually prevent it from shear stress or something if you are putting them in a reactor or something, and also to preserve those things. Yes, synthetic seeds entrapment, correct, you are right, Shweta. Synthetic seeds, and what you call, you can even say, for example, in enzymes and all, enzymes are too costly, so what they will do, they will generally entrap the enzymes inside some matrix, so that prolonged, or some, instead of using the enzyme for one time, they can use it for more than 4 5 times. This will prevent the what you call, enzyme cost to come down, so it will be really helpful. So the question is, the method of cell entrapment or attachment in a suitable matrix is called what? It is cell immobilization. So all these first 10 questions which we have discussed so far now will be having lectures inside in the upcoming week. So need not worry. We will go to the contents of week 1 here. So I think the content of week 1 is more about what? Like what exactly you guys have learned in your week 1? Can anybody brief? What and all you have learned? People started pouring in answers. Okay, okay, everybody is answering so much. So, middle lamella is rich in what? Pectin. Why so? That nobody will answer, huh? So the answer is pectin. Akshaya, sorry, the answer is pectin, which is D. Okay, we will go to the next question. So this is a match the following, like we used to do when we are little, but it will be little trickier, I guess. So can you guys match this uh, co- contents of the column 1 and this side, which is A, B, C, D, alphabets, numbers to alphabets? Can you guys match? I will call out then. So chloroplast is what? The answer would be 1C. Ribosome? No. Chloroplast is what? See all the other 6 ones also, and then you guys form an answer, because there will be tricky answers in there. 1D, okay, chloroplast is absorption of light, correct. Peroxisomes, peroxisomes, 2I, microbodies, correct, Sashank. Xylem, 3B, B is pressure flow hypothesis. Is it so? Xylem is what? Xylem is cohesion theory, correct. Plasmodesmata is what? What are plasmodesmata? They are pits, correct. What is, where does phloem matches? Phloem is 5 is G, which is transport of proteins. No, does phloem do that? Transport of protein? Phloem is actually pressure flow hypothesis. Galvin cycle is Rubisco. Okay. Okay, now what is cohesion tension theory? Can anybody explain? You have all answered correctly. Movement of water through xylem. Okay. What is pressure flow hypothesis then? Yes, yes, somebody was answering. Okay.
Which pulls the water upwards from the root.
Okay.
And allows more water to be filled in the root.
Okay.
And hence water flows through the xylem.
Okay. Correct. And what is pressure flow hypothesis?
Ma'am, the water from the xylem, due to the higher concentration of sugars in the phloem,
Okay.
diffuses into the phloem,
Okay.
which causes the transfer of sugars through the phloem to all parts of the plants.
Okay.
And when it reaches the sink, when the sugar along with water and the sap reaches the sink,
Okay.
then the water diffuses back to the xylem,
Okay.
so that the water pressure decreases.
Okay.
And this is actually the pressure flow hypothesis.
Yes, yes. Good. Akshay, it's really good that you have remembered all those things. I hope everybody is remembering what Akshay was talking about right now. Okay, guys, good. So, question number 13 is, I think this is very easy. So, the outer protective layers of young plants, the epidermis is primarily made up of what kind of cells? A. Parenchyma. Yes, it is parenchyma. Okay, parenchyma. Give me some two sentences or 2 points about parenchyma cells.
It is thin-walled cells.
Okay, they are their shape longitudinal or what they are? They're hard or what? Polygonal or rectangular? Okay, okay. So you guys learned about the dark circle, light circle, everything, right? In the first week. Okay, Vishnu Priya has already put up the answer. So Vishnu Priya and others. So what does this mean? Reaction center chlorophyll of photosystem 1 is called P700. What is the probable reason? The answer is pigments in the photosystem absorbs light at a maximum wavelength of 700 nanometer. What are the other photosystem? In photosystem 2, which one is the reaction center?
650. 680 nanometers.
680 or 650?
680.
Okay. So plants will overcome photorespiration by utilizing dash as its carbon dioxide fixing enzyme. What is that enzyme? How does plants overcome photorespiration? But first, what is photorespiration? PEP carboxylase. So the answer is PEP carboxylase. What does PEP means? What does it mean? What does PEP means? Why does it called PEP carboxylase? Phosphoenol pyruvate carboxylase. Okay. So what is this photorespiration about? Where does it start? What is involving in that?
When photorespiration occurs when oxygen binds with, this binds with oxygen
Okay.
instead of carbon dioxide.
Okay.
Because the amount of carbon dioxide decreases, hence Rudisco's affinity towards oxygen increases.
Okay.
Which leads to the formation of toxin.
Okay. So this photorespiration is good to the plants or bad to the plants?
Bad.
Bad. So it is a bad thing to happen. That's why it overcomes it by utilizing its PEP carboxylase. It comes from which cycle? Your Calvin cycle, right?
Calvin cycle.
Yes, yes. Good. In C4 plants, the 4C compounds are broken down into carbon dioxide and phosphoenol pyruvic acid in the dash where it was broken down. D. Who is nobody else answered? B, C. Each one is giving each so many answers. C, C. Yes, the answer is bundle sheath cells. Yes, Kavita, it is C. So, what are C4 plants? What are the 4C compounds? What are C4 plants first? Tropical plants. The first stable compound is a 4C compound, 4 carbon compound. Okay. So what they are called? Like they are tropical plant to escape from photorespiration. They have adapted something. Correct. So some plants which have adapted to live through the photorespiration and in tropical climates like desert and so. Right. So the answer would be bundle sheath cells And where exactly it's the bundle sheet cells? I want the biology botany people who is here to answer. What exactly is bundle sheet cells? Wreath-like structure surrounds the vascular bundles in the leaves. Not only in the leaves, for everything it will be surrounding it. Okay, specifically in leaves to prevent transpiration. Okay, so endangered and rare plant species can be observed with the help of plant cell technology. Can be conserved with the help of plant cell technology. It's true or false? Okay, true. Okay, so how do we conserve this endangered and rare plant species? We know that plants are technology, but how do you do? Have anybody worked with plants before in your internship or projects or with anything? Okay, many of you are telling micropropagation, ex situ methods. What is ex situ methods? You can tell micropropagation. Yes, micropropagation is there. Somebody told ex situ. Okay, in situ and ex situ methods. What is in situ and what is ex situ? Out of the natural habitat. Okay. What is micropropagation then? Sorry. What is micropropagation? So, endangered rare plant species can be conserved using tissue culture, which is propagating them in vitro to produce plantlets, either to direct or indirect methods. Yes, vegetative propagation, where it involves. Where does the synthetic seed comes into picture? What are synthetic seeds? Somebody answered in the last time, na? Entrapment question. What are synthetic seeds?
Any somatic tissues that is converted to these.
Okay, somatic embryos basically. They are somatic embryos. They can be used as synthetic seeds. Yes, and you can culture them in vitro and maintain in vitro also. And you can then harden them outside and bring it back to their natural habitat. That also is a process. That I think that's what she means. One of your fellow mates mean by ex situ method. What else is there? There is plant breeding strategies is also there. Those also will come into picture. Okay. So give the answer for this. Peroxisomes are responsible for providing energy to the plant by breaking sugar molecules when light is not available to the chloroplast to produce energy. False. Okay, the answer is false. Then what? Where this statement is false actually? So, so who is responsible for providing energy to the plant by breaking the sugar when the light is not available to the chloroplast to produce energy? Mitochondria? Nobody else except Kavita has answered. Okay, we have only two more questions. Rice and wheat are examples of C4 plants. True or false? True. False. Both answers are coming in. False or true? Okay, so the answer is false. So what are then C4 plants? You guys only told that rice and wheat, like C4 plants are tropical plants which grow in desert, kind of rice and wheat grow in desert. What are the examples of C4 plants? I think you guys have cacti and succulents. There are few more others in your lecture, I think. Correct, no? You had a table of C4, C2, a sugar cane, corn, okay. Okay. Rice and wheat are C3, and crops like sugarcane will come under. There were the vegetable names also, na? Nobody remembers. Okay, we'll go to the last question. Cork cells are dead cells that provide protection and prevent water loss. This statement is true or false? True. Okay, where these cork cells will be present? Outer layer of what? Leaf, trees, okay, trees, okay. Where they will be? In the bark. So the bark is a cork cell, right? Stem, trunk, yes, 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 correct. So these cells are they will provide protection as a thick layer and they will reduce the loss of water. Correct, no? Yes. So this as this is the first week of the course, the session is a bit short, and I would like to get your feedback so that the next session I will prepare accordingly for you guys to answer. What kind of questions, or whether you want to discuss on something or you want similar questions to brush up your thoughts on the week two. What you guys would like to do? Or in the week when had you had any difficulties in understanding some topics, you guys can let me know here so that I can take those topics also in the next session. Feel free to tell whatever you feel. Don't worry, I'm not going to grade you on basis of this. The table of C3, C4 plants. Okay, you want that. You want to know that. Okay. Okay, in the next session, I will then show it to you guys. Okay, then what else? So as I'm reactors, based on reactors. So Somesh, actually this reactor portion will be there in your, I think week 3 or 4. I don't remember exactly. The portion will be there. So we will have that a little bit bigger session. Like we will discuss from the basis what is batch. I will discuss here because I can see many of you are from biology background and not in engineering. So we will discuss on that. So good textbooks for plantation culture. Yes. So I wanted to tell you that, but I forgot. So this session is recorded and it will be posted on YouTube, and the links will be shared with you soon, pretty soon. And the textbook details, everything, I will be uploading it in a drive, and I will be handing over the drive link to the NPTEL. So from there you can access the books and lectures, everything. Can we discuss about the doubts from Kavita's message? I missed her message. Can we discuss about the doubts from the lectures, ma'am? Yes, Kavita, you can. We can actually discuss about those doubts here if you want. And please let me know in the discussion forum, you guys, whatever doubts you have, you post there, mentioning that this is from the this. You have any doubts, mean you can ask there also. Still, if you want clear explanation on those, you can let me know. I will provide my email ID here. One second, you can. Or what you can do else is you can mention in the discussion forum, this question is for um, the live session. If you guys mention that, I will be accessing that. So I will be answering in the next, the upcoming week. I will be discussing the doubts, whatever you had in your previous week. So I'm happy to um, clear your doubts in the live session itself. So, but please inform me prior in the discussion forum, or you can mail me in my mail ID. Okay. So, what else you guys need? You guys need plant tissue culture and bioprocess related books. Okay. Some of you, Somesh has asked for bioreactors, and Kavita need to discuss doubts from the lectures. Okay. Anything else? So how does assignment one is coming along? Is it easy? Good. Yes, good. So I think assignments will be pretty easy. So these lectures which are on live are being recorded, and it will be uploaded in the YouTube, and even the drive and the link to it will be shared with you, a drive link form also. So I think maybe by next week it will be shared. I'm not sure when exactly it will be shared. So whatever you guys ask for, the books will be shared there. You can access it there, and the PPTs which I have currently, like the questions, will be also shared there. So if you had any, when you have any preparation exams or something, you can use it. Can you please explain the last question from assignment? Which question, which assignment you are talking about? Okay, so I'm not supposed to discuss the assignment question here until you guys submit the questions. After your submission, maybe we will discuss to it. Yes, it's exam MCQ type. So actually, I'm not part of the exam team, so I don't know about the exam type and stuff. Sorry for the inconvenience, Akshaya. Um, so what else? Anything else? I would encourage you guys to join the live sessions every week. Need not be present all the entire 2 hours. Whenever you please, I know this is a longer session, but you guys can actually pop in any time in the live session and ask your doubts there, get it cleared, so that it will be easier for you also. Okay. So don't think that from the beginning to end it is too long, so I will not be coming there like that. So let it keep us as an interactive session and try to solve your doubts and collect more knowledge. If you guys are done with questions, I think you guys can leave this session. If you have any doubts, please stay back. I'm happy to answer.
போன உள்ள வச்சிட வேண்டியதானே ஏதாவது உள்ள வச்சுட்டு
அது இன்னும் இல்லடா ரெக்கார்டிங் செஷன் முடிஞ்சது எல்லாரும் அனுப்பிட்டேன் இது ஒரு 1.5 மணி நேரமா எடுத்தாலும் 2 மணி நேரத்துக்கு அதிகம் இருக்கும்
சாப்பிடணும்
ஏன்
சாப்பிட
சாப்பாடு
Gracias. போகுதுங்களா? ஊருக்கான உள்ள தெரியுமா?
இருடா பாத்துட்டு வரேன். Thank you for joining the session. I hope the session was useful. Kindly check out the, like, if you have any doubts, kindly reach out to me in my mail ID, which is provided in the text box. Also, you can ask the questions whatever you have about this live session questions in your discussion forum, mentioning that it is from the live session held on 28/7/2023. I will be grateful to answer that questions. So thank you for joining. This is we have come to the end of the session. I will be sharing the textbooks and the PPT along with the video lectures in a drive, which will be shared with you in the upcoming weeks, I guess. So you will get those materials in hand. So with this, I end this session. Thank you for joining.